Good evening, everyone. Mm, Welcome back. And we were discussing about how to construct genomic libraries, I think. And we discussed EMBL3A several times, so I'm not going to repeat it again. But the point is, what is a genomic library? It is a set of clones which are representative of every gene or genetic element present in the genome of the test organism. So we will learn a few more of these in the few more, or at least one more method of how um, the genomic library is made. The other one is called as lambda charon, and it is also based on lambda phage here, and it's called lambda charon 4A. It is a replacement vector very similar to that of EMBL3A, but it's not as complex as EMBL3A. Here, we would take the genomic DNA and cleave it with a mixture of HEA3 and ALU1. I think both of them are 4-base cutters. So we will actually have very partial digests, so we can get overlapping fragments like this, something like that. And then we are going to do size fractionation. Anytime we say size fractionation, what it means is something like pulse field gel electrophoresis. And then we will collect those fragments that are about 20 kb. And then we would methylate it to block EcoR1 sites. How are we going to use? We would have used EcoR1. M is for methylase here. So if you just said EcoR1, it is an endonuclease. So all the EcoR1 sites of the fragments are methylated, something like this. And then they are blunt end ligated through to EcoR1 linkers. I hope you see why now we are actually methylating the fragment with MEcoR1. The reason is we are going to digest or use EcoR1 as cloning site. So if you want to do that, if we are going to add linkers for EcoR1 on both the sites, and if these sites were not methylated, and upon addition of methylase, it would have, upon addition of EcoR1, EcoR1 would cleave here also if it is not methylated. So to prevent those things, methylation is used to not allow further fragmentation of the genomic DNA. And then there is also preparation to be done on the lambda charon 4A as well. We just take the vector and allow the cohesive ends. I hope you remember that the DNA of the lambda phage has overhangs like this. These are about, I think, 12 or 16 bases. They are complementary, and they can ligate and form the something like this. That is what is referred to as the cos site upon ligation. Otherwise, these are the complementary overhangs or cohesive ends. And this is treated with the EcoR1, and then we are going to get three different fragments. One which has the cos site, and there are two internal fragments. By using size fractionation, we can remove the smaller fragments, and what we are going to get is a lambda phage DNA along with the cos site. This is the cos site. Now we have the fragment where methylation is done here, and we have the overhangs which are of EcoR1 overhangs, which are obtained by adding linkers and then digestion. When you say blunt end ligation, we would have used T4 DNA ligase, right? That is, anyway, in general, too, T4 DNA ligase is used majority of the times. So we have the fragment prepared and processed, we have the lambda phage or lambda charon also processed, and now we are going to use these to make ligate these two ends. And then there are several possibilities of occurrence, and this is one, the ideally this is what we would have gotten. I hope you remember the word for it: concatemer. Sorry. Mm, that would have been the concatemer. So you now realize, why do we have cos sites here? Why were cos sites included? Cos sites are important for in vitro packaging. And as long as this distance between here and here is somewhere between 35 or so to about 53 or so kb, then it gets packaged and makes infective phage particles, or which those that can be used as phages. So lambda phage, you can think of it approximately about 40 or so kb, or sorry, 20, total is about 28 kb. And now total is 48 kb or so, lambda phage normal genome. You can assume this to be about 20 or so, or 25 or 30. And the fragment that we have done by size fractionation, we have taken 40 kb. So it is about approximately 50 kb. So it can get, because the distance between the cos site and the cos site is what matters. And it would fall somewhere around 50 kb and can get efficiently packaged into the phage particles. And then we would have selected it on a host E. coli. I hope you can see how much different these two strategies are. The lambda charon is a pretty straightforward, simpler one, whereas the previous EMBL3A is complicated. And with complication or sophistication, we have the application of so many different excuse me, so many different factors, and our job is to learn the tools of it. And therefore we are actually, and that is why we are interested in learning that one. Okay, so the next part is going into the applications and the advantages, disadvantages. We can learn those things. It is there's a simple one. The first one is about, in the previous days before the advent of next generation sequencing, we would have used this as a common thing. Even now, I think people use it as complete genome sequence. That is the first one, and they use these to generate the find out the functions and regulatory sequences, and if there are any mutations in the cancer and so on. Say first, if there is if we take a tumor, cancer, and we would have taken or isolated the DNA from it, and then we would have gone for genomic libraries, and then checked sequencing probably, and then figured out what are all the types of mutations that might have led to the formation of this cancer. That is how we associate the gene or the modification, the change to the consequence, or the what is the cause or causation, and or the cause and the consequence, right? Our job is to be as particular and as correct as possible when we are describing the causation, or a link between a cause and a consequence. The next one is cDNA libraries, and then after we finish this one, today's topic mostly is about cDNA libraries, complementary DNA library. It is complementary DNA is the simplest way to describe it. In terms, the way we describe genomic library or gene library is a set of clones that represent every fragment of DNA present in a genome. There are something else, carbon-Clark formula, and there are probabilities and so on. We are not going to discuss as much of it. So cDNA libraries, if I have to describe or define in the same way, it's a collection of cDNA inserts. It's a set of clones too. Only difference is it is a set of clones representing cDNA of a cell in that particular state, which I hope you understand. This you have to keep in mind. There is genome, and genome gives rise to transcriptome, that is the cumulative of all the RNA that are expressed, and transcriptome will result in proteome, and proteome is the whole of all the proteins expressed at that time. So these are not equal. Okay, genome um, genome is not equal to transcriptome, and transcriptome is not equal to proteome, because genome has not only the genes but also the regulatory sequences, right? Promoter, terminator, and so on. Transcriptome, and there are, say, if there are 100 genes in the genome, not all of them are expressed in every condition. They're expressed in certain states, certain differentiation lineages, and not in others. So maybe around 50 of them are transcribed, and that will make the transcriptome. Whereas, as opposed to what were 100 genes, only 50 of them might have been expressed. And out of those 50 transcriptome, the RNA that are produced, maybe only 40 of them have been translated and produced into proteome. Therefore, genome transcript is not equal to transcriptome, and transcriptome is not equal to proteome. When we make a gene library, we are taking into account all the genes as well as the regulatory sequences and also the intergenic sequences that are present between the genes. In transcriptomes, when we go for transcriptomes, we are only looking at the genes that are expressed in that particular condition or state or treatment or in differentiation lineage. So that is why I had to mention it. Okay, so it's a good way of trying to remember these things, that genome is and transcriptome can vary. So cDNA library would mean the cDNA of all the RNAs, especially those that are encoding the proteins, are it is a set of clones containing all the cDNAs that are expressed in that particular condition or context. So first thing is, we have in the genome library would have just isolated the DNA and then fragmentation, restrictionization, and other things. Here we are supposed to get the mRNA. So the logic is, as you know, in especially that of eukaryotes, there are some things you have to recollect those. Poly A tail on the 3' side, and on the 5' side you have a G cap, that is of the eukaryotic mRNAs, right? So we can use this poly A tail to isolate the mRNAs, mature mRNAs in eukaryotes. So the mixture of RNA will have some of mRNA will have the polyadenylation, and there are rRNAs, tRNAs, which do not have these poly A tails. So we could use a column taken made of, just a second. A column can be made, and the column can have stationary phase and the mobile phase. In the stationary phase, we are going to put these oligo DTs, deoxythymidines like these. Okay, and we can pass the mRNA, total RNA. It's not at mRNA, it is total RNA can be allowed to pass through it. And those that have polyadenylated tails, they will get because of hydrogen bonding, base pairing, they will bind here like this, and the rest of the RNAs will pass through. all RNAs that are non-coding RNAs, including small RNAs, tRNAs, rRNAs, and so on. And later, so this is the same when represented in the principle of it, where now you have the A and T's. This is on the stationary phase, and the mRNAs are bound to it. And then after elution of the RNA, we could wash, I mean, not after the passing of the non-coding RNAs, and then we will wash the column and then allow the, by changing pH or salt concentration and so on, we can disrupt the base pairing between them, and then the mRNA will come out during the elution step. That is how we isolate mRNA. So what we have now is mRNA, and then we need to make cDNA. I think we have discussed a few of it in the previous somewhere, and we are going to see that in more detail. mRNA is the plus strand, and from here we need to get double-stranded cDNA, which is the same information except that this should be in DNA, whereas the other one is RNA. And the second one is mRNA here is single-stranded, and we should get double-stranded DNA, right? So once we can use the same principle of how we have used for purification. As I said, mRNA is this 5' G cap and the polyadenylation on the 3' end. At least we know one sequence. We don't know the other side, right? We don't know the what is the sequence, because different mRNAs will have different sequences, and we are supposed to clone all the cDNA. It's not just one particular mRNA. So what is common among all of them is that the common thing is the poly A tail. So we can design a primer with oligo DT, that means multiple T's, use it as a primer, and add reverse transcriptase so that it will transcribe. And reverse transcriptase is an RNA dependent DNA polymerase. It will for information, sequence information, it will depend on the RNA, and it will polymerize DNA. That is how we can get a single strand, first cDNA strand. Okay, that is one strand is made. We still need to get the second strand as well. So the DNA that is made is base paired to this RNA. We need to get rid of the RNA, and we can use NaOH. I think I already described why RNA is fragile, especially it is because of the 2' OH. Right, this is where the phosphoester bond is there and so on, and you have, this is 2' OH, this will be 3' OH, and RNA has hydroxy 2' instead of deoxy. So in alkaline conditions where you have higher concentration of OH or less amount of H plus ions, it might, this H, it gets dehydrogenated. So we end up forming the hydroxy anion, which will cleave this DNA. The phosphate, the backbone of the DNA gets broken down. So that way, by using alkaline conditions, you can degrade the mRNA, and what we are left with is single-stranded cDNA strand, or first cDNA strand. I think that is the correct way to say. Then we can add DNA polymerase, and DNA polymerase should require a 3' OH for polymerization to occur. Usually, the 3' end of this mRNA, the first cDNA strand, can form a hairpin loop like this. So this hairpin loop can be used by DNA polymerase, and it will add more and more dNTPs, making a double-stranded cDNA. It is still not double-stranded. If you look at it closely, it is one single-stranded, intramolecularly hydrogen bonded, right? Because this is the phosphate backbone is continuous here. And then the hairpin is usually something like this. The hairpin would usually be something like this, right? So these are some of the bases that have not base paired, and they are kind of in this local area, they're kind of single-stranded DNA. And we can use an enzyme called S1 nuclease, which is single-strand specific, and it will digest the DNA. It will break up the DNA now. And what was single-stranded intramolecularly folded, you can call it now as double-stranded DNA like this. Okay. So once we bring it, we can always add linkers, or we can polish them and then clone into any of the vectors. While we study about cDNA library, we don't necessarily speak about the vector as much unless really required. So you can assume how the linkers are attached and then cloned. That will be a regular process like what we have done. We will discuss about some vectors where they are important to discuss. Okay, that is how we can develop double-stranded cDNA. That's the important part of it. There are problems associated with this. One second, it's loading. There are problems with this. It crashed. Give me a minute, please. Here we are. So there are problems with this because the first thing is this is the mRNA, right? From this might somewhere here assume that there is the start codon, and here somewhere there is stop codon. And when it forms this hairpin loop, maybe the ATG was here, assume that. And we are digesting it with S1 nuclease. That means the 5' end of the some of the codons or the part of the gene is actually lost. The second, there is also other problem. Not every mRNA would have formed this hairpin loop. Some of them would form, some of them may not form. If they do not form, then probably we will not get the second strand, right? I'm trying to highlight all the possible problems so that we will, when we look at the other strategies, we'll be in a good place to or understand why those are, how to solve those problems. Okay. So we identified two problems at least. Not all of them will have this hairpin loop, because of which some of the mRNAs might not be converted into double-stranded cDNA. The second thing is we are using S1 nuclease, because of which we might actually lose some of the genetic information from the 5' side of the gene or the cDNA. So it is useful. It's giving some information, but we are missing. It's not ideal yet. So here are some of the earliest method, or what we have seen just now is the general idea. So here is a method based on PBR322, cloning cDNA. We'll make the processing of the mRNA using oligo-DT, reverse transcriptase, alkali treatment to degrade the mRNA, and then the hairpin loop, and we would have used DNA polymerase, DNTP, and then second stranded S1 nuclease. Whatever we discussed in the previous one, it's the same one. S1 nuclease treatment. Now we have double-stranded DNA, and then how do we clone is a question. The simplest way, if you remember the strategy of homopolymer tailing using terminal transferase, we would have taken PBR322 and digested with PST1 and treated it with terminal transferase and GTP, DGTP. And for the cDNA, double-stranded cDNA, all the collected ones, we are using terminal transferase with CTP. So it is as simple. They can base pair, and because of which, and then we transform the construct like this into the host. In the host, the gaps will be filled up. Host repairs all the gaps, and we will have a PST site. Okay, and then we now have a plasmid based libraries, the plasmid based cDNA library. As I discussed before, the same problems are here because we are following this. Essentially, we are following the same protocol of using the hairpin loop to generate the second strand. And to make the single stranded into the double stranded one, we are using S1 nuclease. These are the two problems because of which the cDNA library could be very useful, but it's not complete. There is also another issue when we want to clone. I hope you remember what is meant by directional cloning. Why are we constructing cDNA library to start with? I thought we will discuss at the end, but cDNA library is important for several reasons, especially if you want from eukaryotes. From eukaryotes, their genes have a lot of introns, right? There are interperson intervening sequences like this. And when it forms the primary transcript, it would have formed something like this with the introns. Upon removal of the introns and splicing of exons, we would have gotten the exons. This is the mature mRNA with poly A tail and so on. If we have made a genomic library or gene library and we want to express or produce this in E. coli or other bacterial hosts, it might produce, assume it will produce the mRNA as well, the primary mRNA. It's not yet processed. And the splicing of exons would not have occurred because E. coli does not have machinery for splicing of the exons. So it doesn't work. So that is one of the important reasons why we actually make cDNA library. We are taking this one, the mRNA, and then based on that information, we are making double-stranded cDNA. And that cDNA we are cloning. And once it is cloned and you put it in E. coli, it will be able to produce a protein because there is no problem of exons anymore, right? So these are the reasons why we are doing it. And if I have a vector like this, okay, I'll go to the previous slide to explain this here. Assume this is the vector that I have. I'm trying to clone it here, and I hope you can see it better now. I will try to write, this is the promoter I have, assume that. And is there any scope for directionality in this? If I am cloning the cDNA library in an expression vector, that means it has a promoter and the whichever has to be expressed should be put in proper orientation. That is, it should have the start codon here and stop codon here. And assuming this is the promoter. But because we are using homopolymer tailing here, we do not have any control about whether it is getting integrated in this direction or it is getting in the or integrated in the opposite direction. So if there is a 50-50 chance for anything to happen either in this orientation or in this orientation, we don't want that because we will be missing about 50% of the cDNAs that might have been cloned here. So the objective here is trying to clone it in proper orientation, in this orientation. If you want to clone it in the orientation, we should have had say, for example, I will take this as echo R1, and here I have sal1. So the fragment that I want to clone in here should also have this way. On one side, it should have the echo R1 overhangs. On the other side, it should have sal1 overhangs. Then if there is any cloning, because echo R1 and echo R1, they will ligate something like this. And we have a directionality. We have a promoter, and the promoter will, this is the start codon, and this would be stop codon. And the expression transcription will be fine, and then translation too. So how do we achieve one side EcoR1 and one side SAL1? That is the interesting part of this particular um, strategy. I included this in for discussion just for that reason. Okay, it's a nice way of how you can also think about. This is all about thinking, right? Some scientists has thought well and come up with a strategy, and that is what we are learning. So here we have mRNA and synthesis of double-stranded cDNA. Everything is the same using reverse transcriptase, clinofragment, using the hairpin loop, and so on. We are not addressing all the problems in one go. We are going step by step. And in each case, there is a logic, there is a purpose, and how the scientists might have solved it. And that is what we want to learn. So here, what they do is before addition of SAL1, they would have added linkers, SAL1 linkers. Okay, so SAL1 linkers cannot bind on this side because there are no ends. It is one single-stranded DNA. Here is a hairpin loop, so it cannot bind here. So it will bind only on this side. And then they would have treated with S1 nuclease, and the hairpin is lost, and then you add EcoR1 linkers. So the EcoR1 linkers are added here, here, and also possibly on the other side, but it doesn't matter. When we add SAL1 and EcoR1, you will have cuts here, here. SAL1 will cut here, here. EcoR1 will cut here and here. What are we left with then? We are left with the fragment on one side having EcoR1 overhangs, and on the other end having SAL1 overhangs. Then we can ligate into a vector that is something having like this EcoR1 and SAL1, right? So that we can achieve this directionality of the cloned cDNA fragments. I hope you understood this, and the intellectual part of it is somebody has thought about it well, and this is the part I love in this particular strategy. When and how to do it? How can we achieve adding one linkers on one side and other linkers on the other side? If we have added SAL1 linkers in this position, not here, it would have added on both the sides, right? And then EcoR1, if we added, it would add on all the sides. And then, so strategically, the people have arranged it in such a way that the linkers are on the different ends. You have different linkers. Okay, that's an interesting way of how people have done that step. Okay, sorry, I think it is still okay. Crashed again. Sorry about it. Let's see. Oops. Okay. And next, there are, as I highlighted some of the problems that already are existing. Here are some that will allow us to solve some of the problems. Okay, so let's see. First thing, let's enumerate all the problems that we have. One of it is using S1 nuclease is bad because it is leading to loss of some of the information from the cDNA end, 5' end. And we need to have directionality. That is another issue. We need to have also of the hairpin loop is not a best strategy for formation of the second cDNA strand. Okay, depending on hairpin loop is difficult, or sometimes if some mRNA doesn't form this, then the cDNA will not form. That is some of the problems. We also have some other problems is if we say there is an mRNA, some mRNAs may be broken, right? Some of them may be broken like this. We don't want to clone these broken mRNA. We want full length mRNA. So that is another problem that we would usually have. We are not going to solve all of them today. We will repeat and then slowly we'll solve. But these are some of the practical problems that will come, and that is why we are learning about the different strategies that are available. And what we have to think about is the way the scientists thought about it. We don't need their solutions. We want how they have thought, the logic of it. And there is one more problem. When we use reverse transcriptase, I will put it here. When we use reverse transcriptase, and we add, say, oligo DT, assume that this is complete, and the reverse transcriptase is synthesizing. Say if the mRNA is very long, then the reverse transcriptase, the processivity of reverse transcriptase is not as much. So sometimes it may also cause incomplete reverse transcription, especially if the mRNA is very long. Incomplete reverse transcription. If we happen to make a clone of it and double stranded, we are getting incomplete cDNA. That is also, we don't want to have those in our library. So these are some of the problems that we have when we are constructing cDNA libraries, and we have to pay attention to it. There are protocols, not all of them solve all the problems. Okay, some will solve some kind of things and others may solve the other ones. I will also gently remind you, I think I have asked you to also look at lambda zap vectors. I hope you have looked upon it, and I hope you are trying to do your best learning for it. Okay, here are some of the methods that, one of the method at least we'll discuss, and that is improved. So oligo, this is the mRNA we have, and then we treated it with reverse transcriptase. One second, yes. And then we added DNTPs, and yeah, we got the first cDNA strand. And we want to first, the problem that we don't want to use S1 nuclease. If we don't want to use S1 nuclease, how will we synthesize the second cDNA strand? That is the problem. Right? So here is a protocol that's nice too. Here they have synthesized the first cDNA strand, this one. They're using reverse transcriptase and as usual steps. And then they have treated it with terminal transferase, and they added dCTP. So what does terminal transferase do? Anywhere it finds free 3' overhangs, 3' OH like this, it will add whatever nucleotide is supplied. So it would add CCC here and also CCC here. It is adding even to the RNA also. All it takes care is cDNA, and terminal transferase, if you have to describe it, it is a template independent DNA polymerase. So it just adds bases to 3' OH. So once we have added it, so we know the sequence of one side of the because it is poly A, we have added this primer, so we know one side of the sequence. Now we have added using terminal transferase, we added poly C, and we have treated with alkaline sucrose gradient, so the RNA is removed, RNA gets degraded, and using gradient or centrifuge, we can get rid of the degraded products, and we have recovered the cDNA. I hope you can see that on one side we are aware of the sequence, and on the other side also we are aware of the sequence, because that is what we have used, right? And then because we know the sequence that we added here, we can now synthesize a primer which is poly G, like oligo G, oligo DG, right? Deoxyguanosine triphosphate. And then we can synthesize the second cDNA strand. and then we can insert into vector with homopolymer tailing or using linkers, right? We wouldn't necessarily take up linkers because we might have to digest the linkers with restriction endonucleases, and they may cut also within the reading frame of this. So we would want to probably use adapters instead, or maybe homopolymer tailing. Okay, this is coming at the expense of directionality. We are not having directionality. This protocol is not solving directionality. It is rather solving the issue of not having to use S1 nuclease. We don't want to use S1 nuclease. That is the problem it has solved, so that we are not losing any of the information that is present on this site, right? We are able to synthesize the second cDNA strand by using oligo G, as is given here. Okay. This is basically, I will just summarize here. I thought of describing others, but I'll not go further probably. Sorry, there is something playing out. Yes. Yes. Okay, I'll stop here. If you have any questions, we have 7 minutes or so. If you have any questions, we can discuss. Otherwise, I would want to, did we already discuss about the assignment? So there are two parts to the assignment. The first part is writing, and the second part of it is the oral presentation of it. So both of them are important skills in scientific communication. Okay. Oral presentation is kind of easier because speaking is normal. But of course, you should have the content, what to speak. We are all born with a language and we can somehow speak about it. The writing is the tough one. And that is why majority of the time or since childhood, we get used to copy paste or cut paste and so on. Okay, we don't want any kind of copy pasting or cut pasting in this case. That's why I was very specific about plagiarism. So read as many as papers possible, and first thing you can do is write a table of contents, and then you will get an idea. Or the best thing is, I will put up describe this, and so you can probably follow it. Choose a topic. Any topic that is having to do with some kind of genetic manipulation is fine. And if you're working with animals, it is better. Otherwise, I don't actually care about it much. Choose a topic and try to keep the topic very specific. Don't take general topics. Try to take it very specific, and then read some 3 to 4 reviews on it that might be published or related reviews that on that topic. And then you can also improvise based on them. You write a table of contents, and then you slowly build up the body of the write-up. This is the difficult part. It's not an overnight thing. It will take a lot of time reading, and after you build the table of contents, you have to read a lot of other research papers, which you can find probably from the literature, from the references of these reviews. Read them and synthesize a body of the review, and then you can have a conclusions also. If everything goes good, we can try to publish it. At least we can try. There is nothing wrong. I want to take you from the level of writing to the level of publishing, the process of it. Whether we'll succeed or not is irrelevant. I want to train you, or at least give you an experience of what it means to write about, learn a topic, write about it, and go through the process of publication, submitting, and so on. It is a very nice thing to do. If you can do it, publishing will take time, but that's all right to do that. Once your this much work is done, okay, when you have written the body and so on, you make a PPT of your work. Use diagrams from or figures from the internet and make a PPT and record a video of you presenting what you have written about. So I didn't give two different separate topics. I gave you so that you will have a better understanding. If I told you to write on one topic and present on one topic, it's twice a burden. So instead, I'm making you or asking you to write on the same topic and present the same thing. So your confidence will be better. And when you're doing both these things, your clarity of understanding will be better and you will gain confidence in scientific communication. That is the objective of this particular assignment. Okay. This is for grading. Please pay attention and do as much as possible. Try to start early, and you can, if you want to discuss about the topic, you can always WhatsApp, and then I'll probably respond in time. Okay. If you delay until the last, then we are going to lose a lot of time and I may not be able to help as much. We are quite a large group as well. So this is the general procedure I would like you all to follow. Any questions?
Sir, is it individual only, sir, or
Yes, it
teamwork
is
is also?
individual. No teamworks, individual.
Okay.
Okay. Yes. Any other questions?
No, sir.
Okay. Yes. Take care and have fun then.
Thank you, sir.
Yes. Thank you. Bye bye. Thank you, everyone. Thank you.